Okay, good morning everyone. I am Rohit Chandrikapure and today we are going to discuss about the DNA. So first of all we have to know what is DNA. DNA, it is the building block material of the living organism, every living organism. Basically every living organism having their own character. Like if we take the example of human being, so we have our characters. If we take the example of plants, plants having their character. For example, plants having leaf, root, stem, like that, right? So every living organism have their character, and these characters actually controlled by the factor that is building block element, and that element it is the DNA, right? So it is what it is also called the genetic material. Why it is called genetic material? Because it carries the genetical information from one generation to the next generation, and that's why it is also called genetic material. Now it is a very important, so we have to know all about that. So we will discuss in brief the DNA. So to discuss in brief, we split the DNA into two subtopics. One, it is what the chemical composition, that means what are the chemical components are, means chemically composed by what elements are there. And the second one is the structure, that means what is the morphological structure of DNA. So here, the first it is the chemical component. So basically the DNA it is composed by mainly three main components. One, it is the sugar, then second is the phosphoric acid, which is called phosphate, and the third one is the nitrogenous bases, right? All these components, that means sugar, phosphate and nitrogenous bases, they are combined with each other and they form the unit of DNA, and this unit it is called the nucleic acid. So DNA it is composed of the smaller unit, right? Everything in the world or everything in the universe, it is made by the smallest unit, right? Like that, our DNA it is also made by the smaller unit, and the smaller unit it is called nucleotide. So this nucleotide it is containing by sugar, phosphate and nitrogenous bases. So please, next. Sir. So next. Okay. So the first and very important component of the DNA, which is the sugar. So the DNA it is having a very important, that is the deoxyribo type of sugar. This deoxyribo sugar it is basically the group of ribose, means it is a member of ribose group, ribose sugar. Ribose it is containing 5, means it is the 5 carbon containing compound which is having formula C5H10 and O4, right? Then the next is the phosphate. It is the inorganic component of the DNA, and because of that component, basically the DNA it is having acidic nature. Next. So by this three component DNA actually construct this type of structure. Basically DNA it is having double stranded structure, that means having two strands, and both the strands are bind with each other. So both the strands, that means both the strands bind with each other with the help of nitrogenous bases are there. Means one strand of DNA bind with this another strand with the help of this nitrogenous bases. Again, this is this process when the one strand of DNA bind with the another strand. This process it is called base pairing, and this base pairing it is very specific, right? Why? How this it is very specific? So if the one strand it is having, means basically the nitrogenous bases again classified into two major groups. One it is the purine and another it is the pyridine. Again this purine further classified into two subgroups, that is the adenine and guanine, right? And pyridine it is further classified into two other types, that is the cytosine and guanine and cytosine and thymine. So basically structurally both, that means purine and pyridine, both are structurally different. Basically the purine it is double stranded, while pyridine it is the single ring structures. Purine double ring structure and pyridine it is the single ring structure. So one strand having, if one strand having adenine, so adenine pair with guanine, and the cytosine, adenine pair with thymine and guanine pair with the cytosine. So again this is pairing, that is called base pairing, and this base pairing it is very specific. In between adenine and thymine, two hydrogen bond is there, that means they are shared with two bonds, and guanine and cytosine they pair with the three hydrogen bond. Next. So these are the nitrogenous bases. So basically nitrogenous bases means mainly grouping into two main groups, purine and pyridine. Purine it is classified into two types, adenine and guanine, and pyridine it is classified into cytosine and thymine. So next. Okay. So it is all about what chemical composition. Now we will discuss about the structure. That means the morphological structure of DNA. So basically, this DNA, it was proposed by the structure of DNA. It was proposed by Watson and Crick, right? Watson and Francis Crick. According to them, basically DNA it is having two strands, and both the strands are paired with each other, bind with each other, and they are in because of they have paired with each other, they forming the ladder-like structure, and both the strands are twisted with each other, right? Now by twisting they forming a structure, that means they turn 360 degrees and they form a structure that is called groove. So the groove it is called a complete turn, and this complete turn it has two turns, that means they having two turns. One it is the larger and another it is the smaller. The larger turn it is called the major groove, and the smaller turn it is called minor groove. If we talk about the measurement, so major groove that is it is near about 22 angstrom long, and the minor groove it is about 12 angstrom long. That means the complete groove it is having total 34 angstrom long. And in a 34 angstrom, total 10 base pairs are there, right? That means in a 34 angstrom, in the length of 34 angstrom, total 10 base pairs are there. That means in between two adjacent base pairs. That means if we talk about the distance of two adjacent base pairs, so they having the 3.4. That means 3.4 angstrom. It is the distance in between two adjacent base pairs, right? Then then after each base pair or each nitrogenous base are turned at 36 degrees Celsius. That means total 10 base pair in a groove they turn 360 degrees Celsius. That is called groove, right? And with the help of this groove we can measure the length of DNA, right? So that is the structure of DNA. Thank you.